human diet has a number of fresh produce, such as fruit and vegetables, which are consumed raw and have bacterial populations on the surface, some of which can be pathogens which are harmful to the body. However, little is known about the diversity of produce-associated microbial communities or the factors which influence the structure of these communities. The aim of this study was to distinguish the bacterial communities found on the surfaces of a variety of different fruit and vegetables at the point of sale. Both Leff and Fierer focused on addressing two questions. The first was how the bacterial community organisation varied amongst the different produce types, and secondly, did the farming technique, i.e. organic farming versus conventional farming, have an impact on the bacterial communities found on the produce surface, which is then consumed by the end consumer. In this experiment, Leff and Fierer chose a large range of fruit and vegetables from 3 of the local supermarkets from Boulder, Colorado, USA. These samples consisted of 11 produce items, 9 of which had organic and conventional labelled range purchased as well. Overall, there were 215 samples collected. These samples consisted of lettuce, spinach, grapes, mushrooms, sprouts, strawberries, apples, peaches, peppers, and tomatoes. Bacterial samples were collected from each of the produce samples by the use of sterile cotton swabs or sterile rinse water. The bacteria were then collected from the rinse water and poured to 0.2 micrometer filters by the use of vacuum filtration. A range of different techniques, a number of which involved the analysis of 16S rRNA, were used by the research team to determine the differences in the microbial community composition across the sample items and the differences throughout the bacterial community composition between the different farming practices and the produce types. They found that there were differences in the taxonomic richness on the surface of the organic and conventional labelled produce, and this depended on the
As Figure 1 shows, with the C representing conventional labelled produce and the O representing the organic labelled produce, the organic labelled peppers, strawberries, peaches, grapes and mushrooms had a lower bacterial taxon richness than the conventional labelled counterparts, while the spinach, lettuce and tomatoes had significantly higher bacterial taxon richness in the organic labelled samples, while Figure 2 displays the different families of bacteria found on the surface of the samples. As you can see, the family Enterobacteriaceae was abundant in all of the samples, with bean sprouts, spinach and strawberries containing more than 50% of the bacteria found on the surface of the samples. The next abundant families were Bacterial and Oxalobacteriaceae.
Leff and Fierer suggest that due to the abundance of the family Enterobacteriaceae, that this family may play an important role in the ecology of its host, and may possibly be related to promoting growth and protecting its host from diseases. Leff and Fierer also stated that it is difficult to establish certain factors accountable for the differences between the bacterial communities on different product varieties. However, conclude that is probably due to several factors, such as the variation in storage, transport and handling of the produce. Therefore, it was concluded by Leff and Fierer that the differences in farming techniques and the produce types did have an effect on the diversity and composition of the surface bacterial communities. However, they did point out that their findings did raise further questions. They also state that further research is required to uncover the factors which determine the differences in bacterial communities regarding produce type and farming technique.